Hi, so today we're going to be discussing gel electrophoresis. Gel electrophoresis is a common lab technique used to separate DNA fragments according to their size. And it involves electrical current, which is kind of the foundational principle which gel electrophoresis is based upon. And it is used to compare different samples with differently lengthed DNA fragments. So gel electrophoresis is a very useful tool, and it is used in numerous lab and real-world applications, which we're going to be exploring later. There are also many different types of gel electrophoresis, but today, for our purposes, we're only going to be discussing the most basic and widely used process. Okay, so the materials we need for gel electrophoresis is agarose gel, which we're going to be talking about a little bit later. We need the actual plastic gel electrophoresis box, which holds the gel and the buffer solution. We, of course, need our DNA samples, which is the reason that why we're performing this in the first place. We need electrodes, power source, wires, little alligator clips, which is kind of what this gel electrophoresis is based upon. We're going to need a buffer solution, and we're going to need ethidium bromide and a viewing device, which is what allows us to be able to see our gels afterwards. So to prepare the agarose gel, agarose is a helical matrix comprising of many pores for molecules to pass through. So the structure of agarose itself is very intertwined. There are a lot of holes. So big DNA pieces will be able to pass through slowly. It'll kind of have a hard time passing through, while smaller DNA pieces are going to be able to pass through quickly and more easily. Agarose gel is usually made by dissolving an agarose tablet in some water or gel in some water. For our purposes, the agarose gel has to be a liquid because we need to pour it into a mold to be able to fit the shape that we need. So in order to load the samples, which basically means place the liquid DNA into the little holes that we made with the agarose gel, we use a micropipette in very small liquid amounts, and we place the wells and the DNA on the cathode end of the gel. And the cathode end is actually the negative end. The DNA, well, we'll discuss this later, but this part of the procedure is very important conceptually. So the buffer solution is placed in the gel electrophoresis chamber around the agarose gel, so you kind of just fill it up with the liquid. And the purpose of the buffer is both to carry the electrical current, because that is very important for gel electrophoresis, and to maintain the pH, which is why it's called the buffer. Oops. Okay, so in order to run the actual gel, we need to activate the electrical field through the power supply, and the current runs the buffer, and the current will move the DNA pieces to the positive end. So if you guys remember before, the cathode is the negative end, and we place our DNA at the negative end first because DNA actually has a negative charge from its phosphate group. So it's charged slightly negatively. So since the positive end is at the other side of the gel box, since negative and positive attract, this DNA that's negatively charged will be attracted to the positive end and move towards it. So that's kind of how the gel electrophoresis works. It's taking a while to load. Okay, so each sample is separated into component macromolecules based on its size. So that basically means that the bigger pieces will have traveled less, while the smaller pieces will have traveled more. And usually before we run this DNA, in order to kind of split up the DNA, we use something called restriction enzymes, which cuts the DNA at very specific places. So based on these cuts, it separates the DNA into smaller and bigger pieces, and we kind of see how many smaller and bigger pieces there are, and like compared to the different DNA samples with the gel electrophoresis, because gel electrophoresis allows us to see the differences between the size of the fragments of the DNA. So ethidium bromide or another staining solution is placed, and a UV light box will allow us to visualize what's on the actual gel. So afterwards, we will record the different bands and compare the different DNA samples. So we can kind of see lines below each of the wells to indicate like whether the DNA fragments are there. And this calculation for how many base pairs differences there are using gel electrophoresis is based on a logarithmic scale, which is pretty interesting. It's not linear. So that's all I had to say about gel electrophoresis today. That should give you a basic overview of how it works, its purpose, etc. It's used a lot in forensic applications to compare different people's DNA samples, which is pretty cool. So I hope you guys learned something, and see you next time.